drug therapy. The topics to be covered today are creatinine clearance and hepatic clearance of drugs, the mechanisms of renal clearance, and introduction, objectives, and steps involved in individualization of drug therapy. First of all, what is creatinine clearance? It is one of the most important parameter in drug therapy decisions and is given by the formula UV divided by P, where U is the concentration of creatinine over a certain period of time in a measured volume of urine V, and P is the serum concentration of creatinine. And creatinine clearance can be determined using the Cockcroft and Gault equation. This equation overestimates the creatinine clearance in females, and the estimate has to be reduced by 15%. This can be easily done by simply multiplying by the value obtained from the formula and then multiply by 0.85 to get creatinine clearance for females. This equation is not suitable in pediatrics because muscle mass proportion. Renal elimination of drugs can be through four major mechanisms: glomerular filtration, whereby the small molecule compounds or the drugs are filtered through the glomerular filtrate; renal tubular secretion via pumps like P-glycoprotein; renal reabsorption by non-ionic diffusion of the unionized forms of the drug; and active reabsorption. Hepatic clearance for a drug is given by the product of the liver blood flow and hepatic extraction ratio. Hepatic extraction ratio is the intrinsic ability of the enzyme to mobilize a drug and is also known as intrinsic clearance. And most drugs have a large hepatic ratio or a small hepatic ratio. Hepatic clearance is sensitive to changes in liver blood flow due to congestive heart failure, liver disease, or due to drug-induced changes in liver perfusion. A high hepatic extraction ratio drugs have the capacity to metabolize drug is very high, and hepatic clearance is mainly a function of liver blood flow. The intrinsic clearance does not change much when protein binding, displacement, or enzyme induction or inhibition occur due to drug interaction. The examples of drugs with high hepatic extraction ratios are lidocaine, morphine, and most of the tricyclic antidepressants. Coming to the low hepatic extraction ratio drugs, in these cases, the intrinsic clearance does not change much when liver blood flow is decreased secondary to disease or cardiac diseases. Liver disease or cardiac disease, and valproic acid, phenytoin, and warfarin are the examples. Next, individualization of drug therapy is a multi-step process whereby the first step is the selection of the drug, followed by the estimation of the initial dose based on the target levels, and then loading dose and maintenance doses are calculated. The therapy is begun, and then the therapy is assessed. This assessment of therapy can be through assessment of the pharmacological response in the patient or by therapeutic drug monitoring using plasma or serum drug concentrations. After assessing the therapy, if required, the dose may need to be refined to provide the desired therapeutic effect, and then the adjusted dose is given, and again the therapy is assessed. In case the assessment is still not desirable and it may still require dose adjustment, the dose is again adjusted, and this cycle is repeated until satisfactory therapeutic response is obtained. Objectives of individualization is primarily optimizing the dosage regimen, and to aid individualization, a drug must be made available in dosage forms of different strengths, and the number of doses strengths which can be available depends on two major factors. One is the therapeutic index of drug, and second is the degree of intersubject variability. The small therapeutic index drugs have a greater intersubject variability, and more number of doses and strengths are required. Individualization is based on the assumption that all patients require the same plasma concentration range for therapeutic effectiveness, and the steps involved in individualization of dosage regimen are, first of all, the estimation of kinetic parameters in individual patients, and evaluation of degree of variability is the first step. Then attributing this variability to some variable characteristics, for example, hepatic or renal disease, age, gender, weight, etc., and then design the new dosage regimen based on the data.